One. Hello! Welcome to the Microbiology Journal Club. This week this is our news show. We're going to be talking about the latest microbiology news and some of the interesting papers that have caught our eye.
Mm.
I'm Fazal Lam, and this is my
And
co-host, Danny Chan.
we're happy to be here. We've been here for a while now. This has been like, I guess, one of our anchors through the pandemic. And it's a nice one because we get to try to keep up with the news, and particularly through the eye of diving into the data. We both did our graduate studies in infectious disease microbiology, and this was sort of a good way for us to try to learn what was going on. And so this is our off week, or so we call, where we go through the news articles and we try to choose a paper. And then in the following week, we'll dive in and we'll actually go figure by figure and try to understand what's going on.
Yeah, and so I guess this week we can there's been a lot of news this week,
Hmm.
and we're gonna be talking about some rabid minks, I guess, and some big vaccine news, and we're going to be going through a couple of interesting papers on viral evolution, patient outcomes, and what's been happening with patients, and a couple of other things, just new treatment ideas and new things that we're learning about the virus.
Yeah, absolutely. And our format, I think, we like to encourage people to engage with us. If you have comments, you can leave it in their YouTube channel. You can tweet to us. We can choose the papers. You can ask questions about the stuff that we're looking at. But yeah, I think we should dig in.
Yeah, okay. So the first one we're going to be looking at is the mink studies. So
Mhm.
I don't know if you've heard about this story, but Denmark have gone crazy for mink. They,
Well, they were already crazy for Mink.
yeah, apparently, the number of mink in certain areas of Denmark outnumber the numbers of humans,
Mm,
which is incredible. And another incredible thing is that mink can get coronavirus.
yeah.
Um,
We
so,
actually, this is like a long line of different things that can get coronavirus, right? We've learned
mm,
that it's your similarity to that ACE2 receptor that seems to predispose another species, right, for getting it. And so we know hamsters have a similar one. That's why we use it for the animal model. We know mice do not, and so that's why we have to sort of manipulate them before they are model
mhm,
ready. Ferrets are close enough that they can get infected by the same strains that we get infected with.
yeah. And
Or
there was
sorry,
that
not
paper we
ferret.
looked at.
Oh yeah, ferret. Yeah,
Yeah,
but
ferrets. Yeah,
and mink.
ferrets can.
Yeah.
Ferrets and mink. And there's a paper we looked at briefly, in one of these overview episodes, I think 2 weeks ago, where we
Hmm.
found there's a huge host range. It could even infect some species of whales. That
Yes,
kind of.
whales. I think we're in the mix as well.
Yeah.
Yes.
So it's
I mean,
so
I think
there's
maybe a nice thing about that is we don't have like large colonies of whales that we have close contact with. Um,
yeah, no, not anymore. uh, yeah. So the
so
the
mink
big
are.
thing.
Oh, sorry. I was just gonna say mink
Yeah, no,
are
you
used
go
for
first.
the fur industry.
Yeah.
And so that's one of the reasons why there's so many mink around.
Yeah, and it's especially big in Denmark. So in about the 5th of November, Denmark reported that there were 214 human cases infected with a SARS CoV-2 mink variants. So
Hmm.
these are the variants that seem to have evolved in mink, and there are multiple clusters. But the thing that we're really concerned with this one cluster they call cluster 5 of 12 patients where they're
Hmm.
infected with a specific strain that had four different mutations that seemed to make it less susceptible to say the antibody response. So,
I mean, they
so
didn't. I don't think there's just the concern that it could be less susceptible to the
yes,
antibody response, and I guess the cluster is important because it is on the spike protein, right? That's where we have
yeah,
most of our neutralizing antibodies towards.
yes.
Hmm.
So yeah, they came up with this announcement, and they're saying they were very worried about this mutation, and they basically started to suggest that all mink should be culled, and I think that's still being discussed, but it seemed like a very drastic, and it kind of ambushed us with this news because we weren't really aware of this.
Yeah.
There really wasn't much data available on this mink outbreak. Then suddenly they've disposed quite a lot of new genomes of mink
Hmm.
out there and so they've come out with some more data now that's coming out in dribs and drabs over the past few weeks that it's not more severe, it's not more transmissible, but they are concerned about the antigenicity. So I managed to find there's no data out there, but I did manage to find a preliminary paper, a paper that's basically uh, being made as we speak. It's still
Yeah.
being written, but
Like it
I managed to get
probably,
my
I sense that it could be something where the authors put it up so they could share with their co-authors, right, the version, and so people could talk about it. That's what I'm getting the sense of looking at it, because it's very not polished state. Like the references aren't
yeah,
finished. Like it says working paper as
yeah,
well,
it's a
like to be
working
clear.
paper.
Yeah.
And so they've got like a pic. So they've put up some data showing like where the mutations are happening. So the one of the concerns is this mutation Y43453F, which is in
Hmm.
in the spike binding area, which again
Hmm.
could be. It suggests that for Mink that helps SARS-CoV-2 to bind more effectively.
Yeah.
But then there are a couple of other like mutations in other places that could potentially uh, cause some problems.
Yeah, so I think what I like, I'm really glad that you found this because it really, for me, it fleshes out like that what the who is announced, right?
Mm,
Like the WHO is saying, oh, we're worried about this because there's this cluster of mutations. And you're like, well, what does it even, like, you don't get any of those details.
mm,
And then you can
yeah.
like look at this, you can see, okay, well, they're all in the spike protein. And people, they're hypothesizing in this paper that, oh, it might help it spread better in mink, right? So maybe
Mm,
this idea that it's like, oh, it's spreading in the mink population now, like all these mink are getting it. That's like a whole other petri dish, right? To play around with in the genetic space of a virus.
yeah. And they have some preliminary paper in this data. Sorry, preliminary data in this paper where they look at say that how the titer of this strain. So they call it Delta FBI, and they find that like initially it doesn't seem to grow very fast, but then as it goes on, it tends to like it tends to go up much higher. So there's this concern that it produces a higher level of viral titers, but
Hmm.
the data isn't all in on that yet. So
Yeah,
again,
and
this
that's
is
and that's cell culture as well, right?
yeah,
So,
cell
mhm.
culture in good old Vero cells,
Yeah,
and
but I
then
mean, I
they
think this is it's an interesting thought when you think about the public health perspective, right?
yeah,
Because what is it worth to kill all these mink? I mean, it's a huge hit to those people who make their livelihood off of
mm,
mink, but right, there's this sort of unknown variable where we're trying to contain a global pandemic, and we would like to remove as many unknown variables as possible from this equation. And one potential one that I feel like they are acting on here is this reservoir idea and the sort of idea of evolution in a reservoir and
mm,
being able to come back into humans.
yeah,
And so, like, importantly, in the WHO report, they say, oh, like the people who got infected with this mink strain, it was both people who work with the mink as well
mm,
as people who are in the community, right?
yeah,
Showing that it's not just like it's only a danger for mink farmers here. It's like, here's an example of the viral spread going first into the minks because they didn't give it to us, right? And then the minks giving it back to us and that making its way out into the community. And for everyone's peace of mind, it may be better to just shut down this sector of the economy.
yeah. I mean that Y453 mutation is was ended up being most common variant in that specific area
Hmm.
of Denmark. So
Yeah.
you can understand that. Okay, there is a concern that could this be the new D614G?
Yeah, absolutely. Yeah. I mean, I think it's great that we primed ourselves to talk about this mink thing, right?
Yeah.
Like there's this like we can understand a lot from genomes and it takes a long time to get us to to understanding like what the true impact of that is. And so if it is possible to prevent some of those things from happening, right? If we know the evidence is all the pieces are there, right, to have the emergence of something, maybe it's better to take precautions. And I guess importantly to say here is this is why viral surveillance can be very important, right? Like I
Mm,
think after, maybe 1 or 2 years after we've cleared out a lot of the issues that we're having with SARS-CoV-2, like the conversation should refocus again on surveillance, right? And
mm,
like, uh, I mean, this is maybe a little bit newsish, old news, but the
yeah.
U.S. like had divested from a lot of surveillance initiatives during
Mm,
this time because of the former, I guess, current but soon to be former president, his stance on like international relationships. And
mm,
it's really important to have those relationships intact so that like sort of global viral surveillance can continue,
mm,
because this is something that, like for the initial jump from bats into humans, or like bats into intermediates into humans, or who knows, I don't remember the full story there.
Hmm.
This is the type of work, right? And like, here's an example of the work during the pandemic paying out, right? Something, a preventative measure, perhaps.
Yeah, and up here I've shown the graph that basically kind of elicited the reasoning for all this culling because they took some convalescent serum, some serum from people who were infected with SARS in like the south area of Denmark, and then
Hmm.
they tested like whether it could neutralize the this new virus, and they
Hmm.
found that actually it's that if they you're looking at like people who had so they group their respondents into three categories: people who had a low antibody response, an intermediate antibody response, and a high antibody response to the SARS-CoV-2. And what they found was that the people in those first two groups were not protected against this new variant. Apparently, that
Hmm.
the antibodies were less good at neutralizing, and
Yeah.
so that I guess was is the main reason for this kind of concern. This like
Yeah,
idea that this new strain it means that people who already had SARS could get reinfected with this new strain, and also people who have got vaccinations could also get infected. So, in order to head that off, this is what the reason
absolutely.
for all this
Well, it
culling.
doesn't it doesn't say necessarily about if you got vaccinated you could get, right? Because we don't really know what antibodies are in this mix, right?
True.
But yeah, for sure, I think that the concern would be that even people vaccinated would have the same response. Yeah, I mean,
Yeah.
I'm glad that they took this action. I think I feel sympathy as much as one can economically, right, for
Yeah.
people who have like who work in these industries, but at the same time, like I mean, it's interesting, right? Because I think mink farms are also around the states, right? They're not just in
Yeah.
Denver.
I was
And
just about to say that
yeah.
like about 12,231 mink have died in mink farms due to SARS-CoV-2.
Hmm.
So, and there about been like 11 outbreaks in different states in the US.
Yes.
So it's something to that I guess.
Yeah, so just okay, the other, I mean, I'll say it like I feel like maybe the US doesn't have as good of a test and trace like system. And so like this story that Denmark has, it could be happening here as well in the
Yeah,
States. Yeah.
it could be. I mean, there are other countries as well that have been affected by some things. Like Italy had an outbreak on a mink farm. Spain had like a outbreak that resulted in about 7 people getting infected, and then they
Hmm.
decided to take quite strong precautions against it. And I'm pretty sure there are there are also cases in Sweden as well. So this
Hmm.
is like something an international kind of thought, and there are other like concerns because again we've got like zoonotics, we've got people working on livestock farms. We've got people working in meat
Yep.
processing factories early on in the outbreak who are very concerned about this SARS-CoV-2. So this is
Hmm.
something so something to keep an eye on in the future.
Yeah, absolutely. Yeah, and I'm glad that this work came out. It's like it is concerning, but I also feel like because of the way the action was taken and like these announcements, it's like people are aware of this potential issue, and in those respective areas, we're hoping that people are taking the right steps to mitigate this risk.
Yeah, and so
Yeah.
so I guess we'll move on to the other big news. So we're going to go back to the SARS-CoV COVID-19 vaccine Olympics,
Oh
where
yeah.
yeah. So
I
again,
feel like we've been taking a break from the Olympics because there's just everyone's in their trials, right? And like
yeah,
it was head down, like going through the trials. But I guess some of these trials are halfway or more than halfway done.
yeah.
And so we've been teased with some teaser data.
Yes. So BioNTech and Pfizer released like a press release about giving the data from their interim analysis
Mhm.
of their trial. So
Yeah.
an interim analysis is essentially like where they the kind of cutoff point where they the earliest cutoff point where they can figure out whether their vaccine doesn't work or not.
Hmm.
So yeah,
Yeah.
like they so
So like
so
the
for this
study
one,
has been like let's say they did all these statistics, right, to figure out why the study had to be as big as it is, right? That's the main point, right? Or like the main
yeah,
point of a clinical study. Like all the by the end of the study we should have an answer.
yeah,
Right? And so people use these interim analysis to kind of check up on how a study might be going, right? Like we haven't fully hit statistical significance and everything, but we're trying to see like yeah, we're trying to get a sense of where it is. Um, that's the last thing I think you said.
yeah. Okay, so we're waiting for people to to so that they had 40,000 people. Of those, they estimate 1.3% of
Hmm.
those would get infected over the next year. But
Hmm.
since they're following people about 7 days after their first infection, they've been paring that down. So they're actually waiting for quite a significantly small percentage to appear in that short time period. So they
Yeah.
estimate that the case accrual they'll get in the earliest instant for their interim analysis would be about 137 people, and or that'd be like when they will know that there is definitely going to be an effect of greater than say a certain amount. Um,
Hmm.
so that's when they will know it's effective. Um,
Yeah.
but the but and in this case they've got it up to 94 people out of 164. So that's
Hmm.
about 50% of the way to getting that full like
Yeah.
accrual. And what they found was that 90% of those people were in the group that weren't vaccinated, and only 10% were in the group that were vaccinated.
Yes.
So that really strongly suggests that the vaccine does work.
Hmm.
So
Hmm.
so theoretically, basically, if every single other person, if all the other cases are from are in the vaccinated group, that would still show a 50% effectiveness because they
Yeah.
so that's why I feel like they had the confidence to go out and say, okay, we can definitely say that our vaccine is effective because at this point it's met our
Absolutely.
minimum standards.
Yes, yes. Yeah, so like yeah, I mean the funny thing is it's like very much like what the states went through with their election, right? And like waiting for that last bit of data. This is a very similar approach from the statistics point of view, right? They're sort of like they know that the margins here, like it could be worse, right? It could be not as good vaccine, right? If as the data continues to roll in,
Hmm.
but in all likelihood, it is somewhat, it has some effect efficacy.
Yeah.
Yeah.
So, I mean, I'd caution against people saying that it is 90% effective because at this point it is too early to tell. And
Right.
I've had enough so I've had done vaccine experiments in my time, and I've had that hope in me where like, oh god, these it looks like they've all cleared it, and then it turns out when you
Yeah.
do the failure analysis, it's not quite that level of significance. So there is this thing
Mhm.
called the decline effect that can happen for really outlier results
Mhm.
because you get an outlier result and it'll eventually go back to the mean. And this
Mhm.
is something that I would say looks like an outlier result because I've learned not to have hope in 2020. So I'm
So it's very sad sounding. How about you've
I
learned to be gritty with your
yeah, I mean
skepticism,
it's
skeptical.
90s comics up in here. Like it is not the they're just lulling us. This is like George R. R. Martin going, yes, yes, you're gonna survive. Like no, no, you're not gonna get me like that. I know.
But yeah, I think that's a really good point. Like, we're sort of looking at this abstract sort of clinical output, but like, there's so many factors that could go into that that could just bring this figure down, right?
Yeah.
Like to lower the efficacy of this figure.
So,
But again, I think the
yeah.
fact that they're comfortable making this claim does say that this is likely to be at least an effective vaccine.
Oh
We don't
yeah.
know how effective, but effective in some
I
way.
mean, 50% is because the target that for effectiveness what they were going for was 30%. So they was
Hmm.
basically saying that anything under 30%, we're going to say nope, it doesn't work. But now
Yeah.
they can say that for certain that yes, it definitely does work better than doing nothing. And in fact, it could
Hmm.
work even better than that. But that's
Yes.
only if you want to have hope. But I mean,
Yeah, one of the things that this prompted me to do, because, in the vaccine Olympics, people are working so quickly and trying to show to the investors, right,
yeah,
that they're worthy of the gold medal investment of millions and billions of dollars for their country to buy said vaccine. We don't really see a lot of published scientific studies about what's going on,
yeah,
and we're forced to piece together our understanding of what's happening through press releases. And the other thing that I dove into was this clinicaltrials.gov website,
mm,
which is, the U.S. being a biomedical research powerhouse and sort of economic powerhouse that it is, everyone wants to register their studies on this website to show, right, that they know what they're doing,
mm,
and they are doing these studies, and even to help correlate some of the recruiting efforts, I think, as well.
mm.
And so you can go into this website and you can look for these trials, and you can look at like their enrollment criteria and their outcome measures. And so this prompted me to go in and look at the primary outcome measures for BioNTech, and because I'm sort of wondering how much are people looking at PCR results from the nose, from the nares,
Mm,
right, in these people, or are they looking for the onset of symptoms, and like
yeah.
are they looking at hospital admissions? Because the difference in that outcome measure is really telling us like what is this vaccine proven to do, right?
Mm,
Is it proven just to stop disease at some level, right? Like at a certain efficacy, or is it saying that it's stopping the spread of the virus? And what I have seen is that it's very vague.
yeah.
This website is wonderful that it makes them say something about
Mm,
their trials, but they don't have to put like the details that you would normally see
mm,
in a scientific paper. And so, for example, in the BioNTech one, let's see, do I have this one up in front of me? Yes, BNT1B2. In their outcome measures, what they say is uh, ah, participants without evidence of infection before vaccination. And so that doesn't tell us a lot about how those people are,
yeah.
right? Like, are those people being swabbed at a regular basis, right? Like
Mm.
to see if they get it, or is it if they only report some sort of symptoms, they're brought in and they're swabbed?
So
Um,
yeah,
yeah.
I mean, so it's they Pfizer did release their protocol about a month ago. So
Mhm.
I think they might have some more details in the protocol than they did in the. Again, it was one of those issues where I think like one of the companies went out with their protocol and then all of them decided to do that. So
Sure. Yeah, so I guess
it,
maybe I'll do this for in the coming weeks or something. I'll maybe look at some of those protocols, right,
but
to try
yeah,
to get a sense of this.
but yeah, clinical
Because
trials
it's
are
very
like
vague, like,
yeah.
oh,
Sorry, clinical trials are almost deliberately written slightly vague to give researchers some leeway, just in case like their outcomes change or they learn something new about what they're studying. So
hmm.
it is kind of almost a factor of like a lot of clinical trials that are written slightly vaguely. Um,
Sure. And I
but
think another point is that there's this huge investment, right, in going
yeah,
down the path. So like the idea is that phase one and phase two are supposed to are supposed to get us closer, right? To say like, okay, well, we don't have to worry so strictly about some of these things because we know
mm,
they've passed at least these
yeah,
landmarks. Yeah. But I just want to say, like, in comparison, so like another front runner, at least people are saying in the States, is this Moderna vaccine.
yep,
And so their primary outcome measure, they say, first occurrence of COVID-19 starting 14 days after the booster shot. Right. So again,
right,
occurrence of COVID-19, that's hard to kind of pin down. And then the
yeah,
AstraZeneca, that's the Chadox
mm,
vaccine, they're saying on clinicaltrials.gov, um, oh, prevention of COVID-19 in adults, where the participant, I kind of like this, the participant is defined as a COVID-19 case if their first case of SARS-CoV-2 RT-PCR positive symptomatic illness occurs.
right,
So they're actually looking at symptomatic illness plus the positive test.
yeah,
And I sense that when you think about a vaccine, they want to choose studies that are talking about disease.
mm,
And they may not have primary outcomes that talk about transmission, right? Or like actual like positivity of just PCR, because that might be too sensitive. We don't really know like at what
yeah,
level like you're transmissible like with a certain viral load. So like this gives us like a more definite thing. You can
mm,
say like it is gonna reduce the incidence of disease.
yes,
But I just want to throw it out there, right? We'll probably talk about this again.
mm,
That doesn't say anything about if you get this vaccine, will you not be able to get reinfected or transmit it to somebody else? We have these big unknowns that are still
yeah.
hovering over
And
us.
another thing with the Chadox is that they are starting to do challenge studies as well. So I think about a
Hmm.
month ago they've like announced that they are going to do challenge studies. So it'll be
Yeah,
interesting to see the outcomes of those because those will probably turn over a lot faster than the clinical trials because clinical trials are just waiting for people to get infected. And one of the speculations
absolutely.
is that we're going to get a lot more of like interim analyses because of all the big spikes in the US and other countries which means that we're going to get a sudden a lot of cases, which means that we're going to see a more pronounced effect of these vaccines.
Sure, absolutely. Yeah. I mean, that's sort of terrible silver lining, right, of
Yeah.
an impending spike is that ongoing clinical trials are going to be tested, right? They're going to get a better significance because they'll see more cases, right?
Mm.
They put these big wide nets out of like tens of thousands of people, right?
Mm.
And the net is going to catch a lot more cases if the rates are going up. Not a good reason for rates to go up, right?
Yeah.
They were powered given very conservative estimates of spread. So
Yeah.
like we don't need this. We don't need this spike.
Oh.
Yeah.
Yeah, and so I guess we've said everything about BioNTech, but that's not the end of the story. Oh no, because 2 days later we had some news from Sputnik 5, because yeah, they
Because
also
they also
decided.
want to claim
Yeah.
some good efficacy results.
Oh yeah,
Yeah.
I mean they really are going for like the first COVID vaccine ever to be like approved, which again
Yes.
is kind of incorrect because a couple of the Chinese like heat kill
Hmm.
vaccines were approved a lot earlier,
Yes.
and it's amazing that we haven't really been we haven't been hearing much about Sinovac and Sinopharm, but they've been like publishing phase 2 studies. They've been like chugging along, and
Yeah, they've published their phase two. I've heard that different countries have purchased, right, those
yeah,
vaccines, manufacturing rights to those vaccines. So they're out in the world. They're starting.
yeah.
Hmm.
But yeah, so Sputnik V is they published their interim results in a press release on November 11th, where they highlighted 20 cases in which there are 90%. They show like 98% of efficacy or something like that. There's sort of very high efficacy for 20 cases
Hmm.
out of again 40,000, and
Yeah.
so if we do the same calculation with them and saying like, okay, well they need to reach a similar threshold of say 100 cases. They're not quite to the 50% threshold level. So,
Right. Yeah.
but
20 is less than the 90.
yeah, 20 is like yeah, it's about a fifth of the way they
Hmm.
want to of where they want to be. Um, but I'm not going to say it's all bad because again, having so if this is sustained, if they have continuous like if they come with the next one and they still have this level, then that is also quite a positive step. But at
Hmm.
this stage, they really don't. It could go either way. I don't think it will, but I think that they are like kind of treading on a much thinner ice. Um,
Yeah.
but because yeah, I've been there where like, oh yeah, my this first mouse seems to be responding really well to the vaccine. It seems to be really good. This could be work, and then the other's like, nope, and I'm like, oh.
Right.
So it's so yeah.
And then we
Again,
also have then there was also a press release from Moderna,
yeah,
right?
Moderna as well. Moderna has they just said, oh yeah, we're gonna publish an interim analysis at some point. Uh, so they're going to include more than 53 cases in interim analysis, which I'm not
Mhm.
sure that speaks to how effective the vaccine is.
No. Just that they have the data, I think. I think they
Yeah,
want to tell us they have the data. The data is coming. But I think this is just seeing this response in the media, right? Of like press releases coming out to talk about these different vaccine results, giving us the interims. It really speaks to this idea that not everyone, we're not seeing, um, in the rush to get this vaccine out the door, right? The only real people that have to be shown the data are the regulatory
mm,
bodies. If the regulatory bodies are even engaged, right? Because like Sputnik 5, like we're saying, there's some question whether the regulatory bodies were properly engaged in that case. Um,
mm,
but like this is those are the people that we're sort of trusting, right, with this with the green lighting of these things and the broader scientific community, which includes us, right, other
yep,
scientists working in different labs. We only have published scientific research to go off of and these press releases, which aren't always like fully fully fleshed out for us to do a good deep dive on them and to understand what's going on.
yeah. I mean, I almost forgot to talk about the other instance with the BioNTech paper where they were originally going to publish an interim analysis a lot earlier, but then so they in the original plan they were going to publish it at an earlier level cases, but for some reason they pushed it back again. Like Moderna seems to be pushing back on like not. So there could be different ways to interpret that. I know a lot of people are saying that it's a political thing that oh they
Mhm.
didn't want it to interfere with the elections.
Yeah.
Another thing that could be said is that perhaps they didn't get there as perhaps this was a point where it looked the most favorable to them because
Sure.
again these cases appear like almost like randomly, so you get clusters of people going into each group. So again, you might
Hmm.
have only seen this big like jump in cases in the control group because of the most recent jump in cases in the US. So it could have just been that it didn't the actual like differences between the groups didn't look amazing until like just now, and then that's why they decided to publish it. And the reason why Moderna are holding it back is because their cases don't look that great either. But then,
Yeah.
so um, so it's one of those things. So I think
Absolutely.
that
And those that's driven like that idea of like one group not having all the cases they need versus another group having it sooner. That's driven by so many
mm,
crazy variables, right? Of like
yeah,
how they did their population selection, right?
yeah,
And then how does that overlay with the outbreaks that are going across the country? Um, it's actually if people have been following along with us or like have been following along with just news media about like treatments and stuff, this is very similar. There can be parallels in terms of like just study design when we're thinking about like how remdesivir data slowly accrued,
mm,
right? Like they published their end result from this Chinese cohort, but by that time there were so few cases, right? They didn't hit like a certain significance, and they
mm,
couldn't do like the subgroup analysis that then they later did, right, to try to show like, oh, it's only for these very the efficacy is like concentrated in this very early time point of people getting the treatments. So yeah, it's just we are dependent, and this is what challenge studies will try to rectify, right?
yeah,
Is that you don't have to worry about sort of like yeah, just the variance of who's getting what when. Um, the other thing I just wanted to say about challenge studies though is I'm hoping that through the process of sort of ethically flagging like the need and the procedures, right, that they'll take with challenging people, that may also legitimize like more constant sampling, right?
mm,
Like because this is a very important, a very uh, like I guess valuable, right, like opportunity, right, to use human challenge studies,
hmm
then it may also say like, and these people will also have more rigorous data collection so that we can know better what's going on. That's certainly an exciting thing to think about.
yeah, for definite. And yeah, I think that brings us to the end of the vaccine Olympics for the moment. We're still waiting on actual data to be published. Uh,
Yes, basically lots of positive buzz news, but that's coming from the investors. That's coming from the people who are making the vaccines themselves. And we're not seeing a lot of secondary analysis, right? We're not seeing like the scientific community speak about it as much.
yeah. So I guess that brings us to the papers that we might want to select for next week.
Mhm.
So
Okay, the first one we have is we have some from this evolution
yeah.
thought.
So yeah.
Following up, I guess, on our Mink and our D614 stories.
Yep. The circulating SARS CoV-2 spike variant N439K maintains fitness while evading antibody-mediated immunity.
Hmm.
So
Interesting. So this is one of those concerns, I guess, right? This is
yeah.
a a paper that's showing us a potential concern of what mutations could be like. I'm just throwing up the bioarchive now so I can look at this paper.
Yeah. Uh,
Uh,
so it's a
oh,
really big study. I mean, we did we did speak on it a little bit last week because it's one of the multiple mutations that we were like spotted as being quite common in the GSAID network. Um,
yeah. Oh, yeah, that's right. It comes up in the large sequences that we've seen before for this.
yeah, and it's on spike, and I think the big concern here is similar to what the concern with Mink that neutralizing antibodies that neutralize normal like original recipe SARS don't necessarily neutralize this one as well.
Yeah. Yeah, let's see what sort of figures do they have in this paper.
Yeah, let's,
So from a quick glance, we're looking at oh, they're always at the bottom of these preprints.
yeah, they're and they have a
Always at
lot of collaborators on this paper as well. So this was involved in there are multiple people involved in this paper. Uh,
Yeah.
Again, I think like uh, so they've got like a look at the protein and variants where mutations happen, and a cool figure they have is comparing like so they're looking at Scotland and England, and they've got like two different like kind of outbreaks of this strain, and
Hmm.
they've plotted out it happening in Scotland, and then showing like how it's really exploded in England and Ireland in recent months.
Yeah. Yeah, it looks like they're doing that version of the test that we talked about with the mink, where they look at convalescent serum from different
Yeah,
people and use it against the variants in neutralizing um, tests.
yeah,
Okay. Yeah, this is pretty, it's a big paper. It's about a mutation. We've sort of talked about mutation before. I don't know, but it could be very interesting.
yeah. They test some antibodies. They use Regeneron and Ly-Cov as well against it to see the efficacy of those treatments as well. So it does go
Hmm.
a bit further. So it's quite nice.
Okay. Cool.
Uh,
Let's see. We have another one.
yeah.
Dynamically evolving novel overlapping gene as a factor in the SARS-CoV-2 pandemic.
Hmm.
Um, I mean, I brought this one up. I don't think that this one is as in depth that we could look at. This is actually related a lot to at one point, I think I suggested a paper that was saying they did a proteomic analysis of all the different things that could be made. And
Right.
this one is like a sequence gazing experiment where they like looked deeper into the sequence and they're like, oh, there might be another protein here that we hadn't previously looked at.
Yeah, that's interesting.
Oh, and then they go back and they say, okay, yeah, it is made because they do some mass spec as well.
Yeah,
Um,
I quite like Eli papers because they've got things like decision letters, authors' responses, and other sorts of things that they're a bit transparent about. So, uh,
Sure. Yeah. Like choosing a paper from this publication, you get to see a lot of the research process more than you would typically see right from a publication.
yeah.
Mhm.
So yeah, I mean, this and like BMC are quite good with transparent peer review, where
Mhm.
they um, so I mean, it's something we might look at those publications a little bit more, but but again, we follow the stories, so the cool stories is what we want. So that
Yeah. I see. And they're actually trying to take this ORF that they found and correlate it with like just like how they did how they did the mutation when we were talking about D614, right? They're trying to say, oh, look, it's coming up in different amounts, right?
yeah,
Yeah. Interesting. But then they don't have that key piece that we talked about. What's the significance of seeing this of seeing this genetic thing? What's the biological significance, right?
yeah,
Of seeing this genetic change occur.
yeah, yeah.
Okay,
Okay,
interesting. Let's see. We got in patient studies now. We
yeah.
have prothrombotic,
Or
thrombotic.
yeah, prothrombotic autoantibodies in serum from patients hospitalized with COVID-19. So this is
Mm,
actually quite an interesting idea where because thrombuses are like basically clots
plot.
in your blood like that kind of occlude your that block your blood vessels, and
Hmm.
so that could be again that causes that's something you don't want to happen. It, it can lead to
Yes,
like lots of bad things happening to you.
for sure. This is kind of this is this is the patient story of right. This is like the big patient report
Mm.
of something people have been saying about like the endothelial
Mm.
infection, right? Like I think they were talking about all these clotting side of like clotting symptoms that occur
Yes.
with SARS-CoV-2. So here's like a good report like showing us like this indeed does happen. And there has been information that's come out that saying like this is a function of the infection, right? Like it's an interaction between SARS-CoV-2 and endothelial components. I think we even may have spoken about it briefly last 2 weeks ago when we were
Mm.
choosing. There's like there's some like complement. It can touch complement, right?
Yeah.
Like the mechanism, right? These are
Mm.
mechanisms in which you could see these effects occurring. So this is almost saying like there's an autoimmune effect, right?
Yeah,
Autoantibodies.
that actually isn't because it messes with the immune system so much, and I mean there was that paper of it having some super antigenicity or something. That wasn't really about that,
Yeah,
but the idea that like it does cause the immune system to go into this inflammatory overdrive. That
right.
a lot of like diseases do that. So and that's how
So
like
the, it could be the overdrive, right? Like we could try to stitch it together, right? And they could be maybe saying like the overdrive is influencing antibodies that end up having a pathogenic, right? A disease
yeah.
causing effect on us. Yeah.
So I mean, there is almost like so. In this case, it's part of the infection because the good thing about having something that's prothrombotic is it stops blood going to certain places, like the places where your virus is growing, or places where. So again, that can hamper the immune system. It can also, uh, I mean, sometimes it can be done by accident. So I know that there are like various things where it triggers an autoantibody to where like you get sequelae, where the immune system goes into overdrive and just starts to
Yeah, that's how I was reading it. But you were saying it could not, it could also be part of the disease process itself, like the
yeah.
body's adaptation. Yeah.
Yeah, so I know but this is more of a bacteria thing though. So, like
Yeah.
things like super antigens, and they tend to work on a much like smaller scale to help the to divert attention away from the bacteria. I'm not
For
so
like
sure
the
whether
local environment of the local environment of the infection.
yeah,
Yeah.
so in the local environment that can help the bacteria to survive, but if it goes systemic, then suddenly everything goes a lot worse for everybody, including the
Right.
because
Right. Yeah, because it's a heightened immune response now on the for the bacteria and for the patient or for the host. They're experiencing some crazy side effects,
yeah,
some yeah,
yeah,
some widespread infection.
and those can always persist after the bacterial virus are gone. So things are called sequelae, which are like the sequel to your first disease, but you thought it was over, but here's a sequel.
Okay, interesting. Yeah, I
Yeah,
mean, we haven't talked about this whole the whole set of endothelial and clotting related effects of COVID-19, right?
yeah,
We was
and
so that's interesting from this perspective.
they do to
Um,
take it into mouse studies as well in there as well. Um,
yeah.
but yeah, anyway, next one:
Okay.
distinct antibody responses to SARS-CoV-2 in children and adults across the COVID-19 clinical spectrum.
Hmm.
So this,
So this is observation,
yep,
uh, and trying to give us a like a good way of thinking about testing, I guess, right? Like
mm,
better resolution on testing. Like, can we know stuff from testing these antibodies about the outcome of disease and so forth?
yeah.
That seems like what it's trying to tell us.
And it's interesting because like children do get a slightly different disease outcome to adults. They're less vulnerable, so it could be interesting to see
Mm.
what the children doing that adults aren't doing. Well, I mean, and could that inform treatments or could that help people survive? It's
Mm.
it's an interesting place to like fish for an idea.
Yep.
Uh,
Okay.
okay. We have like
Oh, case
two
study.
case study. Yeah.
Prolonged infectious SARS-CoV-2 shedding from an asymptomatic immunocompromised cancer patient.
Right.
Um, I think maybe I'll present this more as a, I mean, I guess there's quite a few figures
Yeah.
in this, but this is just again, a case study.
Mm.
So we're only seeing one example, but I guess maybe it is important because it's in a human
Mm.
and we're seeing evolution inside of an individual. So we've talked about this evolution happening and like worrying that it's going to spread out, right? Into the population,
Yeah.
but this is like zooming in, just saying like in one person that they were able to get many samples from. What did that evolution even look like? What's
Yeah.
going on? Um,
I mean, it's kind of interesting because I know with HIV, the evolution during a patient infection can be very important because it can
oh,
because
it's huge.
yeah,
Yeah.
because it can switch, and so it'd be interesting to see like because again like immunocompromised cancer patient, they're already kind of vulnerable to it, but also
Yes.
like seeing like this it slows everything down, so you can see the SARS-CoV-2 adapt in real time, so it's
Yeah, you can see the dynamics, right, of that evolution play out. I guess like the risk again is like, okay, well, exclusion criteria in a lot of these vaccine trials are being immunocompromised,
Mm.
right? Like these are people that we really have to protect. There may be another reason to protect them because they could be hosts for evolution
Yeah.
and your reservoir in that way for the virus.
And I think the
Okay,
next
anyways.
paper also, well, so the next one is a letter, but it kind of like builds up on that story again with the persistence and evolution of SARS-CoV-2 in an immunocompromised host.
Mm-hmm. Yeah, I mean it's almost the same thing, just not as detailed it doesn't seem. They don't have as much data in this one.
Yeah,
This one is just like they only show this one chart of the evolution. Yeah, very similar actually, I
mm.
would say. Companion papers, these two are.
Yeah. So SARS-CoV-2 in evolution, it doesn't just happen in mink; it happens in people as well.
It happens in people as well. Yep.
Yeah.
And in this case, right, like I mean, we don't, right, like only vaccination and proper protection, right,
Yeah.
of the broader population is going to remove this issue. So,
Yeah. And these patients are also being treated with convalescent antibodies, and I believe like maybe not Regeneron, I'm not sure, but because I didn't read the paper in that. But again, one of the things that Regeneron was quite in their early papers, they're talking about trying to plan it so they could plan for escape
absolutely.
mutants.
Yeah, yeah. Uh,
So again, that'd be an interesting point of comparison between these papers. Um,
Agreed. Okay, let's move on. We
yeah.
got a bunch of treatment papers coming up.
Yep. Design.
Design of a highly thermotolerant immunogenic SARS-CoV-2 spike fragment from
Yeah.
BioNTech.
This is an this is a paper that seemed a lot more interesting to me on because it goes very much into the biochemistry of like how the stability of the SARS-CoV-2 protein and for vaccination and just
Hmm.
it does some early work in guinea pigs, um,
Yeah. Yeah,
but
so this is sort of in the preclinical bandwagon, like
yeah,
we could even say second wave of potential therapeutics.
yeah,
And I guess the development here is focused on trying to make uh, easier to distribute vaccine, right?
yeah,
Because if a vaccine is made with something that's thermotolerant, it means that you don't have to worry about all that cold chain logistics that people are talking about right now with the vaccine. So that's the hope, I think, with this, but it's
yeah.
still in the very early days.
Because I guess everyone's talking about the whole -70 issue for the BioNTech Pfizer vaccine, where
Hmm.
like it
Hmm.
is you have to like keep it in like uh, not liquid nitrogen, but the other one, dry ice to keep it, make
Dry
sure that
ice. Yeah.
yeah. Um,
So yeah, if this stuff comes to bear, if these types of technology come to bear, we may have vaccines. I guess this would probably be because they're designing it. I guess they could still encode it into mRNA. No, no,
oh
because
no, no,
that wouldn't. No, this would be making the protein itself.
yeah,
This would be like the Novavax
yeah.
vaccine.
This
Yeah,
would
the
be very
Novavax.
much in the nanoparticle Novavax vaccine, so it'd probably
Yeah.
be a bit more expensive to do. I think maybe I'm not sure, but
Mhm.
it, so yeah, this is probably not going to be an amazing vaccine candidate, but it's interesting to look at that thermostability because again,
Yeah.
like SARS spike is
No,
already
I don't think we can say that it won't be a good vaccine
yeah,
candidate, but it's like it's this particular one's really far away from it.
yeah, it's pretty
Yeah.
far away right now. But uh, anyway, the next one is quite interesting though: molecular targeting of vulnerable RNA sequences in SARS-CoV-2, identifying clinical feasibility.
Mm.
So this almost is so they take an siRNA approach of like small interfering RNA approach
Hmm.
to 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 battling a SARS infection. So
Yeah. So
uh,
I guess this is like as we're learning more about the infectious cycle of SARS-CoV-2, maybe this is like a totally different uh, candidate for a therapeutic, right?
yeah,
Where you're just saying you can't make this protein of yours anymore, and that's
yeah.
going to stop the infection.
I mean, I think there is even I mean, they're mostly targeting Spike, I think, but they're because I mean, we did those papers where they're looking at how SARS like affects the spliceosomes and how it affects uh,
Yeah,
like ribosomes and all those. So there are lots of other like kind of targets for for like the SARS production that you can use to
sure.
not only to prevent infection, but also to mitigate the outcomes of the cells that are being infected as well. So I think there's
Hmm.
a lot of because siRNA for treating oral infections is a really interesting idea. And
Yeah. I mean, I think there's not that many siRNA therapeutics out there in the world right now. Which again, like always, it floors me that we're having mRNA vaccines coming out,
yeah, yeah, I know.
right?
It's
Yeah, because people.
because yeah, we can with those vaccines we established that we can now deliver mRNAs into cells. So the question is like whether we can do enough
Get
to.
that, that.
Oh, sorry, I couldn't hear you. Broke up there a bit.
Oh yeah, I also had a similar hiccup. I was just saying that what's great is like, yeah, I think you said uh, the fact that we have these mRNA vaccines is showing us that we can get mRNA in, right? We can make cells make the mRNA that we want. This is the same thing here. It's just a sort of a different use of that RNA. Instead of making a protein, this RNA is going to directly grab onto other pieces of RNA and stop them from being translated.
Yeah, I mean, again, looking at the aspects of these vaccines as gene therapeutics as and that kind of potential, because I guess that's kind of what Moderna were about initially, right? I mean, they're about like RNA therapeutics. I mean, that's what um, I thought. So yeah,
For sure, for sure. There's a huge explosion basically of yeah, RNA therapeutics, sort of gene therapy adjacent
yeah,
technologies.
because they're a lot safer than gene therapy because you don't have to worry about like germline integration or potential cancer problems.
Yeah,
It's like mRNA, it's
for
much
sure.
safer. So uh, yeah, so
Yep.
that's what next paper. Uh, ooh,
Another
de
therapeutic,
novo,
ACE2
yeah, de
decoys.
novo design of potent resin ACE2 decoys to neutralize SARS-CoV-2. So essentially throwing a because SARS-CoV-2 it binds ACE2. So if you throw enough ACE2, then it'll just like stop binding to um yeah
Yeah. I don't know if you remember, but in that Regeneron paper, they were using, right, they were trying to throw
yeah
shade on
they were.
this type of therapeutic.
I remember that, yeah. So,
Yeah, they had shown that in their hands, their monoclonal antibodies were much better at neutralizing than some ACE2 decoys.
yeah,
Um, but in this paper, I guess they haven't given up on the
yeah,
technology. They've decided to go in and try to design even better decoys that probably show better neutralization
yeah.
profiles.
I mean, they do cryo-EM to show that this design can bind all three receptor binding domains of a spike protein. So it
Hmm.
does like they do like put in a bit more evidence, but it's again it doesn't necessarily discredit the Regeneron idea because I imagine this was still being done while that paper went out, and they're like, ah,
Yeah.
we'll get
For sure. Yeah, I mean, it would be funny to see if they, I guess they don't, if they did a head-to-head, right? They're like, and we also tried a cocktail of monoclonals and it didn't work as well as our
Well, the thing
decoy.
about these
Um,
decoys, so if you do get mutations like the Mink one and they still have to bind ACE2 no matter what. So,
yes, that's right.
so
The decoys give us a maybe a more robust therapeutic to potential mutations on the viral side.
yeah, so yeah, that's that. I mean, hopefully,
Okay.
and they also talk about like a single intranasal prophylactic dose, like protected Syrian hamsters. So not even like putting intravenal, but just like sticking it in the nose of a, and it.
Mhm.
So that seemed like a quite interesting. Again, they said lethal. Yeah,
Yeah, that's like a Tamiflu type
yeah,
of application, right? That's like
yeah.
a yeah,
So again, that seems like an interesting therapeutic paper. Next one is
for sure.
structure and drug binding of a SARS-CoV-2 envelope protein transmembrane membrane domain in lipid bilayers. So, tr- so this is yet another protein of SARS-CoV-2 that could be a drug target for uh,
Hmm.
whatever we want. So
Yeah, that's
yeah,
E, right?
E
E
protein.
protein. It's the one that we've seen them use as like the subgenomic E is used as counting
Yeah,
genomes.
uh, yeah, and so they basically
Yeah.
produce a structure of it. So this is mainly like a structure paper to look at how it works, and then
Oh man, they're using something strange. They're using sort of NMR
yeah,
structure, right? I see. Uh, this is a structure we haven't talked about before. Strange, like resonance and isotopes doing things.
yeah.
I hate reading these graphs. They're not easy for me to wrap my head around.
Yeah, and NMR, it's because mostly we've been looking at crystallization optical kind of techniques like cryo-EM, but NMR is almost
Yeah,
like a entirely different way of thinking about things. You're looking at magnetic and resonance, so
yeah, you're seeing it like what
yeah,
jiggles
you're
and,
you're
and
banging the protein and making it glow in some electromagnetic magnetic radiation area, and then
yeah, and the way that that radiation comes out is like influenced by the environment of the things around it, and so that can create the constraints that builds you the
yeah,
structure.
but again, interpreting the data takes a slightly different approach to what we're used to.
Yeah, but from the therapeutic side, it's
Mm,
very interesting to think of targeting this E protein
mm.
instead. Um, how would it even? I don't even know how it would stop the. Right? How would it even inhibit the
So yeah,
virus
I think
blocking?
this is, I think the E protein forms like plays a role in fusion or like in
Yeah, or maybe budding? Is it
yeah
in release? Maybe it's in assembly,
yeah
right? Like maybe these particles don't get assembled properly.
yeah
I feel like the only data they give here is in that's all just like that's the
mm,
hypothetical, right, of A that they think it works here.
yeah.
Interesting.
So I guess that's more
And also, I mean, maybe just this can even just be seen as learning about the E
yeah. I think
protein.
that's probably more about where they
Yeah.
go. "Look, here's an E protein," and they're like, "Why should we care about the E protein?" And they're like, because it can be targeted for drugs. That's why." Do you have any
Yeah.
evidence? We could theoretically. Yeah. Okay.
Yeah,
Fine.
maybe. Don't rule it
Yeah. You
out.
don't want to.
Science moves in
Yep.
mysterious ways.
Oh.
Okay, let's see. Also on the docket, we have SARS-CoV-2 neutralizing
Mm.
antibody, LY-CoV555, in outpatients with
Yeah.
COVID-19. So, yet another monoclonal antibody that could have some effect.
Yeah. I mean, this is
I guess.
because we haven't talked about Lykov, but that's the other like main competitor Regeneron at the moment.
Mhm.
So uh, they're made by a different company. I can't remember which one.
This is like they're in patients,
Yeah.
right? So this is we're seeing some real efficacy
Yeah.
data here that we haven't seen previously. Not so much about mechanism. We've sort of talked a little bit about monoclonals
Mm.
before as well. This may be just interesting to
But
point
yeah,
out
this
for
is like
people.
interim
Uh,
analysis.
I will say though, I think I saw a tweet or someone shared with me a tweet just saying like, this monoclonal business, this is like the natural evolution of having tried a lot of convalescent
Mhm.
serum in people. Of course, now like for widespread biotech, right, distribution of these therapeutics, it's much more controlled to give people monoclonal
Mm.
cocktails. Um, yeah. And last, we have inhibition of coronavirus entry in vitro and ex vivo by a lipid-conjugated peptide derived from SARS-CoV-2 spike glycoprotein HRC
Yeah.
domain.
Now this is interesting because you're taking a part of the coronavirus's own spike and using it against it. So that's a bit. So this kind of came out of a
Yeah, definitely. If you thought decoys were weird,
yeah.
right? Like, here's another very strange way of getting at a
Yeah, they
therapeutic.
look at like
HRC. HRC is in the intermembrane, is on the is inside of the
yeah.
virus.
So it's so the idea is it blocks infection by messing with the fusion. So it stops
Hmm.
the actual fusion of the SARS protein S by I think messing with the fusion protein that would usually be inside the spike, but then comes out. So again, this is kind of a confusing like I mean this is something that I didn't really get.
It's hard to imagine. It's hard to imagine just by reading this title what that mechanism would
Yeah.
be. um, but yeah, but apparently they have evidence that it works. It seems like they identify this region and then they do a bunch of fusion assays, right? Or
Yep.
neutralization assays. Um, and then they don't really give us a final mechanism, but it has effects. Very
Yeah,
strange.
this is very strange. So, it's something that I, yeah,
In some ways, I mean, we've never talked about this region. We rarely talk about the S2 region of
yeah,
Spike. right? Because we're talking about S1. That's the one that's outside, flapping
Mm.
around, trying to grab onto the receptor. This is like, for some reason, when you make something on the inside, when you make a protein on the inside, it inhibits what's going on.
Yeah.
Maybe does it maybe get in? No, but they're probably adding viral particles. Yes. Strange. I don't
Yeah,
know.
because I mean, this is literally the injection point, the thing that would inject into the membrane to cause SARS-CoV-2 to
Hmm.
fuse. But I guess having other versions of it in the membrane might interfere with this action, or it might.
Yeah, maybe it interacted. Maybe other parts of the protein interact with this during fusion to complete that
Yeah,
step, right? There's like some multi-process step of fusing. Anyways, so those are everything that we have. We gotta choose
yeah,
something now.
gotta make a
Um,
choice.
yes.
Hmm.
I do like I actually do like that E protein one, just because I'm curious about the E. Also
Yep,
siRNA might be
quite.
interesting to talk about.
Yeah, I'm quite.
Uh, Even decoys. I mean, these are all three different therapeutics, right? That
Hmm.
we've never talked about before. Um, or sorry, we have two different therapeutics that we never talked about before. We have something sort of masquerading as a potential therapeutic, but more about the biochemistry of what's going on. Uh, do you like the prothrombotic autoantibodies? I feel like that's kind
Yeah,
of
actually,
interesting as
I do
well.
think that because again, that goes very much into the clinic, and also it does pair that
Yep.
with mice. Um,
Yeah, we're seeing disease models. We're seeing like a disease process. It's something that has a lot of background
hmm,
that's been out before in terms of like I know like there's all these like blood thinner,
hmm,
right? Like what's the use of blood thinners in the clinic and like what are some of the endothelial effects it could tie into there.
yeah. Uh, so I mean, at the moment, the thing that really is catching my eye is the sRNA and the HAS ACE2 ones. But
Okay. Okay.
I mean, I'm also willing to
I think I'm up for the HACE2 one, I think.
yeah, I am too because
Yeah, the
yeah,
decoy
it does
method.
cryo-EM. It does a lot of it does a lot of interesting things that I wouldn't expect. Um,
Yeah.
uh,
Well, I mean, it's interesting to think about drug design. We haven't actually talked about de novo
yeah,
drug design, right? Like this is very different. Like we've talked about like convalescent, we're
mm,
fishing for things, right? Monoclonals come from the fishing from
mm,
convalescent serum, right? And the fishing of different things like that. Um, Vaccine, yeah, remdesivir that existed
yeah.
beforehand, right? They just had that one ready. Um, yeah, I'm up for that de novo
Yeah,
design.
okay. De novo design of so next week we'll be talking about
Potent and resilient H2 decoys to neutralize SARS-CoV-2.
yes. Yeah, so yeah, I think that's what we're going to be. So if you want to join along in the audience, then the links to all of the papers are in discussion. Everything we talked about, we referenced, and we're going to be also, of course, the thing we're going to talk about next week, we're going to be really diving into is de novo design of potent and resilient HA2 decoys to neutralize SARS-CoV-2. Yeah, we've said it enough times, so hopefully we'll. But
All
yeah,
right. See you guys
yeah,
next
see you
week.
guys next week. Thank you so much for joining me, Danny. It's always a pleasure to have you on the show. Uh, yeah,
Wonderful.
right. Thank you. Right, bye.